Okay, good morning and welcome to my class. This one is for grade 12 STEM students only, and our class is Biology 1. So before I start with our first ever lesson, I just have a few reminders for everyone. So after watching this recorded video, you have to take note that you have to read the reading material. The textbook is uploaded on your LMS. I have indicated the page numbers that you have to read. So after watching this video, you have to read the reading material. And after watching and reading, if you have questions or some ideas are still confusing, take note of those questions, take note of those clarifications because I will be asking them on our live Zoom meeting. So during the live Zoom meeting, I will just address your questions and your clarifications. I will not be doing a repeat discussion of the topic, just those ideas and questions that you have. So take note of them. We will be doing them during the live Zoom meeting. And then I've also included links to YouTube videos that you can watch to help clarify or to help you master the topic. I have also included links for online assessments. Now I have included asterisks here for the online assessment just to clarify that these are not graded and they are not recorded. I have given them for the purpose of you mastering the topic, but they are not graded and they will not be recorded. The purpose is just for mastery so that when I give you the actual exam, you are sure or at least you are confident to get a good score on those assessments. And another reminder, my meeting ID number for our class, if when we do our Zoom meeting, regardless if it's biology 1 or another class, a different class that I'm having with you, I am using the same meeting ID number. That's 391-527-8060. So take note of that. And my password is always science asterisk 87. Take note, science starts with a capital S. There is an asterisk and the number 87. So since I'm done with those reminders, let's start with the first ever topic for Biology 1, and that is biological molecules. So when we say biological molecules, it means that these are the molecules of life, which means that all living things, whether it's humans or animals or the insects, the small insects, we all have these biological molecules. And these biological molecules are actually made up of organic elements, specifically carbon, hydrogen, oxygen, nitrogen, phosphorus, and potassium. So mostly, or all of our biological molecules are made up of those made up of those three. So carbon, hydrogen, and oxygen. Some molecules, such as the amino acids, has nitrogen. Some molecules, such as the nucleotides, have potassium and phosphorus. But they are all similar because they have carbon, hydrogen, and oxygen. So we have 4 different macromolecules in our body, and like I said, it is shared by all living organisms. The first one is carbohydrates, the second one is proteins, the third one is lipids, and then the fourth one is the nucleic acids. So these 4, carbohydrates, proteins, lipids, and nucleic acids, these are what we call as polymers. So when we say polymers, it means it's a long molecule. It's a connected long molecule that is actually made up of the same building blocks. And these building blocks are what we call as monomers. So the monomers are these. For carbohydrates, this is a polymer. The monomer is a monosaccharide. So if I'm going to add one monosaccharide to another monosaccharide, then I am already building a polymer. But the smallest building block of carbohydrates is always monosaccharide. Now for proteins, this is a polymer. The monomer is an amino acid. If I only have one amino acid, then I'm referring to a monomer. But if I am talking about 5 amino acids, then that is already a polymer. Now, the same goes for lipids. Lipids is a large molecule, and it is composed of many fatty acids. So the monomer is a fatty acid, the polymer is a lipid. And then for nucleic acids, the monomer is a nucleotide. So if I'm going to add 5 or 10 nucleotides, then I am forming what we call as nucleic acids. So how do we form these polymers? We form them through the process of dehydration reaction. So when we say dehydration reaction, it means that we are building something from small and we are making them into bigger molecules. So it says here from monomers, which is your single unit, we are going to form larger molecules. So we are adding monomers to another monomers and then adding another monomer to create a polymer. And this process is what we call as dehydration reaction. Now remember that a dehydration reaction always creates a water molecule. So for example, I am going to add one monosaccharide to another monosaccharide. So now I have 2 monosaccharides. That is a polymer. And when I add these 2 monomers, I am going to create 1 water molecule. Now, the opposite is what we call as hydrolysis. Hydrolysis, we are disassembling long molecules or polymers and we are making them smaller. So from big to small. With dehydration reaction, we are making from small to big. But when it comes to hydrolysis, we are starting from big and then we are making them smaller. So we are disassembling them. So the process is called as hydrolysis. And you have to remember that in the process of hydrolysis, we are going to use water molecules. With dehydration, we are creating water molecules, but when it comes to hydrolysis, we are using water molecules. So I'm going to show you how that happens exactly. So for example, this is a short polymer. I have 1, 2, 3 monomers here. And then I'm going to add another monomer here so that I can create a polymer made out of 4 monomers. So the end of this polymer, it ends with a hydrogen atom here, or a hydrogen molecule here. And then this monomer has an OH here. So in order for them to connect or to create a link between these 3 polymers and this 1 monomer, they are going to create a water molecule. So now we created a water molecule and a polymer that has 4 monomers. So this process is what we call as a dehydration process. Now the opposite of that is your hydrolysis. So when we say hydrolysis from something big, we're going to make them smaller. So here we started with a polymer that has 4 building blocks, 4 monomers. Again, one end ends with an OH and the other end ends with a hydrogen. So in order for us to remove 1 monomer, we are going to use water. So we are going to use water here. And then this water will be divided into a hydrogen ion and an OH. So the hydrogen ion will go here to this polymer, and then the OH will go here to this monomer. So we are breaking it down. Now you have a polymer that has 3 monomers, and then you have a separate monomer here. So again, just to recap, the dehydration process, we are starting from smaller molecules, we are making bigger macromolecules. And in order to do that, we are creating water. And then the hydrolysis process, from a bigger molecule, we are going to break them down into smaller molecules. And for that, we have to use water. So now let's start with the very first biological molecule, which is carbohydrates. So when we say carbohydrates, the function of this is it actually provides us with the energy that we need for our everyday activities. That's why if you will remember in the food pyramid, the one at the bottom is always carbohydrates. It means that we have to consume more amounts of these carbohydrates because we use it every day when we breathe, when we walk, when we write something or read something. We are always using energy, and that energy comes from carbohydrates. And carbohydrates it includes monomers and polymers of sugars. And like I said, the monomer of a carbohydrate is your monosaccharide. So this is the single unit of sugar is always a monosaccharide. And then when we try to create larger molecules of carbohydrates, then we are talking about polysaccharides. So the monosaccharide has a formula of CH2O. It is composed of carbon, hydrogen, and oxygen. And the most common monosaccharide is what we call as your glucose. Now I'm going to show you what glucose actually looks like. So this is your glucose. So as you can see, it is only made up of 3 elements, your carbon, your hydrogen, and your oxygen. The molecular formula for glucose is C6H12O6. So let's count. If it has 6 carbons, so this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. So let's count hydrogen. It says there in the molecular formula, glucose has 12 hydrogen. So let's count. Hydrogen 1, hydrogen 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And then it also has 6 oxygen. 1, 2, 3, 4, 5, and 6. So this is glucose. You will see a lot of glucose for biology 1, including biology 2. So take note of the structure and the molecular formula of glucose. So glucose is a monosaccharide. So we can add one monosaccharide to another monosaccharide. And when we have 2 monosaccharides, we call it as disaccharide. So coming from di, it means 2. So it says here that a disaccharide is formed during a dehydration process, which joins 2 monosaccharides. So when we form a disaccharide, we are also forming 1 water molecule. And the bond or the linkage that is formed between the 2 monosaccharides is what we call as your glycosidic bond. So all polymers of carbohydrates have this glycosidic bond, regardless if it's a disaccharide or a polysaccharide or made up of more than 2 monosaccharides. The link between the monosaccharides are always made up of glycosidic bonds. So these are the most common disaccharides that we have. If I'm going to add glucose and galactose, I'm going to form lactose. And lactose is commonly found in the milk that you drink. So lactose is made up of glucose and galactose. If I'm going to add one glucose to another glucose molecule, then I am forming what we call as your maltose. So maltose is a disaccharide. And if I'm going to add glucose and fructose, fructose is the sugar that we find in fruit. If I'm going to add that with a glucose molecule, then I'm going to create your sucrose. And sucrose is commonly found in your table sugar. So again, these are your disaccharides. Lactose is made up of glucose and galactose. Sucrose is made up of glucose and another glucose, and then sucrose is made up of glucose and fructose. So that is your disaccharide. Now let's go to your, let's show how these disaccharides are formed. So this is your glucose, and I'm going to add it with another glucose molecule. So this is a dehydration process. So again, the OH here bonds with this hydrogen ion here, so it creates water. And then in between them, we are forming what is called as the glycosidic linkage or the glycosidic bond. So here, the oxygen here, and connects the glycosidic bond. So again, if I'm adding glucose to another glucose, I am forming maltose. So this is already a disaccharide. Now the same here, I have another glucose molecule, the OH here on the end of the glucose molecule in carbon 1 of glucose molecule. And then I have here fructose, which has another OH on carbon 4. So this OH here and the H here will form your water molecule. And then you have the oxygen forming the glycosidic bond. And then glucose, when we add the fructose, we are creating your sucrose. So this is your table sugar. So now you have your disaccharide. After the disaccharides, there are also carbohydrates that is actually made up of more than 3 monosaccharides of sugar. So these are what we call as your polysaccharides. So we have 3 most common polysaccharides, and these are your starch. The starch is most commonly found in your plants. Your plants have the ability to create their own food, especially when there is abundant sunlight, because they use the energy from the sun to create their own food. Now, your plants, if they have enough water and if they are exposed to enough sunlight, they can create unlimited amounts of food. And they have the ability to create more than what they need. So if they are already satisfied, they already have enough energy or enough monosaccharide for all of their activities, they still have extra glucose, and these extra glucose are stored in the form of starch. So most of the plants that we are eating that provides us or serves as good sources of carbohydrates, such as your rice, your potatoes, your sweet potatoes, and corn, all of those contain starch. And those starch are actually extra glucose that the plant was able to create. So they are just storing it in case they need it in the future. They will be using the energy from that starch. They will digest it back to glucose, and then they will use it to provide energy for the plant. The same when it comes to animals. In animals, we don't have starch. We have what we call as glycogen. So for example, me, I like eating sweet food. I always have a lot of extra sugar in my body. So that extra sugar is actually stored in the form of glycogen. Now this glycogen, for example, today I'm eating a lot because there's a birthday party in our family. So the extra sugar will be stored as glycogen. And then next week, I started exercising, and then I went to work, and then I didn't have time to eat lunch. So in that case, the sugar in my body is low. What my body will do is it will use all of this stored glycogen. It will digest it, and then it will become, from glycogen, it will become glucose. So this glucose will be released into my bloodstream, and then it will be distributed to the different cells. And then the cells will use it to create energy for the different activities that I will be doing. So if I'm always eating extra sugar, my body will store it in the form of glycogen. And in the future, if I needed extra energy, but I haven't eaten anything or I did not input sugar in my body, what my body will do is to convert this glycogen into glucose, and then those glucose will be distributed so that my body cells can create more energy, even though I did not eat anything. Okay? So, yun yung joke nila when it comes to apocalypse, na pag wala ng pagkain, ang matitira is yung matataba or yung may extra storage of glycogen in the body, which is actually true. So, kung payat ka, during an apocalypse, walang masyadong glycogen na nakastore sa katawan mo. After a few days, all of those stored glycogen will run out. So, wala nang source of energy yung katawan mo. You will die. Pero yung matataba, they have a lot or they have more storage when it comes to glycogen. Kahit 1 week or 2 weeks, their body can still digest those glycogen and then convert it into glucose. But eventually, yes, when your body runs out of glycogen to digest and it can no longer produce the glucose that your body needs, syempre, regardless if payat or mataba, you will eventually die. They said that you can survive without food for 7 days before you actually die. But without water, you will die in just like 3 or 4 days. So water is needed other than, of course, your food. So glycogen is found only in animals, such as humans. Starch is only found in your plants. And then cellulose, we find this usually in plants that have a lot of fiber, and it's also found in the cell wall of all plants. So cellulose is not just, it does not function purely for the storage of energy. It's actually, its function is more of the structure that it provides to your plant cells. And later on, when we discuss about the different cell organelles, you will understand that plants have cell walls and they have different, their cells look different because of the presence of this cell wall. And this cell wall is made up of cellulose. So let's discuss that one by one. So again, your starch, these are found in your plant cells, and their primary function is for the storage of extra food or the storage of polysaccharide in plants. And then for your glycogen, it functions similarly to your starch, but it is only found in animal cells. So again, it is storage for extra polysaccharides or extra glucose in your body. And then we have the cellulose. Cellulose is primarily not for storage, but it's more of for structure. It provides structure to the cells because it is found in the cell wall of plants. Now, polysaccharides, again, let's go back to your glucose. Let me go to my Jamboard. Okay, so this is my Jamboard. I have here 2 glucose molecules. Again, let's count. This is your carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. Again, this is carbon 1, carbon 2, carbon 3, carbon 4 here, carbon 5 here, and carbon 6 here. Why am I emphasizing this? Because when it comes to your polysaccharides, they are categorized according to the glycosidic bonds. Now, when it comes to glycosidic bonds, we have 2. We have alpha 1,4 and we have beta 1,4. Now, when it comes, for example, alpha 1,4, in this glycosidic bond, it is indicating that the linkage or the glycosidic bond will be formed between carbon 1 and carbon 4. So if this is carbon 1 of one glucose molecule, it will bond to carbon 4 of the second molecule. So the glycosidic bond will be formed between these 2 molecules. And then here, the OH here will bond with the hydrogen here. Okay, so here. So this is your alpha 1, 4. Okay, so let's see. Let's go back here. So this is what I'm talking about. In your starch, the glycosidic bond is formed between alpha 1, 4. So this is carbon 1 and this is carbon 4. So now we are forming the alpha 1, 4 glycosidic bond here. So for starch, if you will notice, all of the glycosidic bonds are formed downward. It means that it is an alpha 1, 4 linkage. And this is commonly found when it comes to starch. But when it comes to your cellulose, it is actually connected by a beta 1, 4. So again, we are talking about the linkage between carbon 1 and carbon 4. Carbon 1 and carbon 4, and here carbon 1 and carbon 4. But what's different between alpha 1, 4 and beta 1, 4 is that in beta 1, 4, you have one linkage that's going up, and then the second one is going down, the third one is going up, and then the fourth one is going down. So it's alternately going up and going down. Whereas when it comes to alpha 1, 4, all of these linkage are always going down. So when it comes to cellulose, because it alternates between up and down, it's possible to stack this cellulose on top of one another. So if this is going up, the second sheet here will also have something that goes up. And if this goes down, the second sheet here will also go down. So it's possible to put them on top of each other. So as a result, cellulose are always in sheets. They're always stacked up against one another. This is not possible when it comes to alpha 1, 4. When it comes to your starch, all of the glycosidic bonds are going down. So if you put one on top of the other, it's going to be bulky. So these starch are usually helical in nature. So they twist and then they go around one another, similar to what your DNA actually looks like. So again, for your starch, it's made up of alpha 1, 4, and alpha 1, 4 always go down. So because they always go down, they form rods or helical. Whereas when it comes to your cellulose, they are alternately going up and going down. So it's possible to stack them up with one another so they can form sheets, or they are possible to stack up with one another. Okay, so again, polymers with alpha 1, 4 glycosidic bonds, they form helices, whereas your polymers of the beta 1, 4 linkage are always straight because they can be stacked up with one another. They can form straight sheets. Okay, and again, your starch is found when it comes to your plant cells, and they are storage for extra food. Your glycogen is found in animal cells. Their function is for storage of extra food. And then your cellulose molecules, which is made up of your beta 1, 4, they form straight sheets. They are found in cell wall, and their function is not for storage. They are more for forming structures. So that is the end of the topic for carbohydrates. After this, please read the reading material. I have given the page numbers. Just go to the textbook and go to the page numbers and read those pages. They're not really that long. They're like 2 to 3 pages long. And then take note of your questions so that on our live meeting, you can ask those questions and I can test you on your understanding. If you have extra time and if you have extra data, especially if you have Wi-Fi at home, please do watch the videos and answer the online assessments. They will really help you to master the topic when it comes to carbohydrates. And then when you have mastered the topic, you are ready to answer the worksheets and to do the experiments that was given again on LMS. And then after doing that, we will be having a short assessment that will be graded and will be recorded. But do as much as you can to master the topic. Okay, so if that's that's the end of my first class. On our second class, I will discuss the second biological molecule, which is all about protein. Okay.